A vaccine is used to arm our immune system. It contains messenger RNA, which can direct cells to make specific proteins. The messenger RNA tells our cells to produce COVID spikes. These harmless spikes give our immune system the information needed to quickly detect a coronavirus infection and produce the appropriate antibodies. The new antibodies respond once COVID has entered the body. They attach themselves to the spikes on the COVID virus, rendering the spikes useless, protecting our cells and keeping the virus from reproducing.
The candidate vaccine is designed to raise an immune response that will help the body fight off the SARS-CoV-2 virus. It targets a protein on the surface of the virus called the spike. The spike protein helps the virus gain entry into a person's cells, including those in the lungs. Once inside the cell, the virus replicates. It can then enter many other cells in the body. In response to the infection, the body's immune system produces antibodies against the SARS-CoV-2 virus, which stops the virus from multiplying. A vaccine that targets the spike protein can help the body build this defense by simulating an infection without making the person sick.
A COVID-19 adenoviral vector vaccine is an adenovirus vector based on a common cold that has been modified and inserted with the genetic material for the SARS-CoV-2 spike protein. Production starts with assembly of its genetic code. The adenovirus vector is engineered from adenovirus DNA by removing essential genes to stop it being able to replicate, so it can only act as a carrier and not cause disease. Adding the coronavirus spike protein DNA creates the full genetic sequence for the adenoviral vector vaccine. The genetic code is introduced into a producer cell, where it is transcribed and translated to form the COVID-19 adenoviral vector vaccine. The human cell line is engineered to contain the missing adenovirus genes, so that when the vector vaccine is introduced, it can infect the cells and use the cell machinery and missing viral genes to be able to replicate, producing identical copies. Vaccine molecules are also replicated with the division of the cells, and the process continues until the right concentration is reached. The addition of a chemical lyses the cells, bursting them open, and the vector vaccine is harvested, ready to be further tested, filtered and purified before being filled into vials.
Now what we're seeing are actually mutants which are quite different from the original strand. In fact, it's like the syllabus that has been changed. So now when I used to score 95 marks with this standard syllabus exams, I do worse off because it's a totally different mutant, it's a totally different syllabus. But even if we went back to the original strain, the efficacy was only 95%, which means while the 100 people there are, 5 of them will still end up with an illness, but we understand that to be mild. Vaccination changes a severe disease to a mild disease, and from mild disease to an asymptomatic disease. Overall, about 30% of us will have very long-lasting antibodies that will cope, and the other 30% won't have enough antibodies. It will fall with time, or they never mount adequately. And in those cases, all the more, you need to remind the immune system by going for a booster vaccine. When the antibody levels fall to a certain low level, we probably have to go for vaccination again. Booster shots work by jolting the memory of the immune system. We may be able to find good matches of different vaccines that will even exceed what we'll be getting all along with the traditional 2 doses of the same vaccine.